So hello everyone, again, Microbehunter here. Welcome again to another microscopy live stream, to another Saturday microscopy live stream. I'm currently checking again if everything works. Today I would like to do a little bit of something of a microscope comparison. Yes, I can see myself also in the browser. Very good. So yeah, sound also seems to be good. Hi, hello everyone. Today I would like to put a few sample slides again under the microscope, but as you can see over here, I've got a pretty low cost introductory microscope, and I connected my 5 megapixel USB camera to it. I'm just holding up the camera so you can see it on top. Yeah, and I would like to show you some of the specimens this time, not through my regular microscope, but actually through this fairly cheap microscope over here. And you can already see that I put a tick up. I bumped the camera. I put a tick up over here on the side, other side, yeah, over there. This was actually the slide that I was looking for last week. In my mess on my workspace, I could not find this slide because if you count the legs of this tick, you're going to see that there are only 6 legs. Normally ticks have 8 legs, and the reason why this only has 6 legs is because this is a tick larva. So this is a little bit from last week. Yeah, I see that many people have already started to post in the chat. I would like to, if you're new to this format, to this live stream, you can please ask any microscopy related questions that you would like. Please put at the front at Oliver, it's my name, or at Microbehunter, so that I know that this is directly directed to me, so I can ignore some of the other lines here. Otherwise, it's a little bit too difficult to find. As long as the questions are microscopy related, I'm of course going to be willing to answer them. And I did already receive one question right at the beginning of this chat here, and for whatever reason in YouTube, I don't know why, some of the comments disappear after some time. But there was one short question that I would like to answer because some other people also had a technical problem with their microscope, and I would like to answer this first, and then I'm going to actually go directly into the topic that I wanted to talk about today, and that is the problem of a condenser that is not centered. So this was actually a comment, the first comment. Some microscopes, they have those condensers on the bottom. The microscope here on top does not have this. Okay, so that's actually quite an introductory one. But sometimes this one is not in the middle, right? And then people say it's off center, and you can actually see if you look through the eyepiece. Yeah, here it is. If you look through the eyepiece, then you're able to see that you kind of. Yeah, it's not in the center, right? Because there's this little white hole is not in the middle, and it's quite easy to center it. And what you have to do is you have to look for some screws on the bottom side, right? So this is beneath the stage, and you have to take one of those wrenches here and simply loosen the screws. You do not remove them, right? You just make them a little bit loose. There's 3 of them in this case, right? Here, and here. 1, 2, and 3, right? And all you have to do is you have to make them a little bit loose, and then you can center the condenser, and then you can tighten it. And just want to show you that the holes here are actually larger than the diameter of the screws. So actually this has already been designed to be able to have a little bit of a wiggle room so that you're able to center it, okay? Because I know that this was actually the Swift SW380T microscope. That's actually one of the microscopes that I also have been recommending. I think it's a little bit sloppy manufacturing, that they simply put it together, but they did not check whether it's actually in the middle. Sometimes, therefore, it's not in the middle, but it can be really easily fixed, and it is actually intended to be centered. That's why the holes are a little bit larger, right, than the diameter of the screws here. So that is essentially the thing that I would like to simply wanted to answer here, because some people had the problem. So those condensers, if they're not in the middle, you can try to look for some screws here on the bottom, and then basically you make them loose, and then you can center it. And that's it, okay? So I just wanted to quickly answer this here, because there has been a request in the chat yeah, for me to quickly answer that. Okay, so what I'm going to do, and I think now more and more people have joined. Usually it takes a couple of minutes for people to join in. 62 people are watching right now. I'm really happy. More and more people are starting to join in every week. So I'm kind of very happy that hobby microscopy actually is becoming more and more popular here. And I would like to talk now a little bit about those introductory microscopes here, and some of the advantages and disadvantages that they have. And then, of course, I would like to also compare the samples and specimen slides with my other microscope that I have next to me. Okay, so basically, what I will also do every now and then is I will interrupt myself, and I will also answer some of the questions in the chat, even if they're not directly related to the topic, but as long as they're microscopy related, I will answer those. So what I'm going to do now is the following, is I'm going to, I don't know how I'm going to do this, because I have a problem a little bit of showing this here. This here is what I call an introductory microscope, around 100 euros or $100. Okay, some are even cheaper than that, and those microscopes have a couple of advantages and a couple of disadvantages. The main advantage is, of course, that they're pretty cheap. Okay, and these are not toy microscopes, but they can actually be used also as portable field microscopes and so on. Yeah, made mostly of metal. That's the stage in this case is plastic. Of course, some parts are also plastic, but overall it makes a very solid impression. And usually they only have one eyepiece, and I basically took out the eyepiece and connected now a camera over here, USB camera, which is now connected. You can see when I turn this, you can see that the image also turns. It's now connected to the program, to the computer, right? The microscope that I have over here, and that is important a little bit, has 3 magnifications. It has a 4 times, a 10 times, and a 40 times objective here. And down here, if you look down here, there is no condenser, but there's just a wheel that you can turn. However, I consider this wheel pretty useless. There's some opening holes in the wheel here, but they don't do anything, right? So I don't even know why they added this. Yeah, there is a power control here. It's powered by LED. The problem with that is, it's not a problem really, but it's not a very consistent. Yeah, so the light intensity does not change a lot. Yeah, you might be able to see it at the top. Yeah, and it has coarse and fine focus knob, and this screw over here on the top is important for setting the maximum height so that you do not crash the objective into the stage. Yeah, so basically it has all of the essential features, and that's actually one of those, this type of microscope, not necessarily specifically this model, but this type of microscope, I generally say if you're completely new to microscopy, then and you don't know where to start, then just get one of those, get your toes a little bit wet, and then upgrade to a better model later on. Okay, but the question is now is how good are they? And the reason why I decided to make this live stream is because I do receive quite a few emails where people are asking me, well, how good is the image quality of a certain type of microscope? And a little bit the point that I'm trying to make in this live stream is that the image quality itself is not so different from more expensive microscopes. Of course, the better microscopes have a better image quality, of course, but you might be surprised that the actual difference is not that large. So, and in order to prove that, I'm going to now take out this tick here, okay, that I have, and I'm going to put it now into my other microscope, and then let's have a comparison. Okay, so that's kind of the idea. As the first, I don't know if this is such a good idea. This camera is a little bit in the way here. And I just want to show this to you. So what I'm going to do is I'm going to click over here now to the other one. I have to remove this one over here. I put it in there, and then I have to find it again. Here it is. Okay, I have to adjust a little bit. I have to focus a little bit. Yeah, here it is. Let's go up with the magnification. Okay, I'm going to now do bright field so that essentially, yeah, it's a little bit comparable, and that's how it looks like with my Olympus. Here it is. I don't know, you cannot, I have a problem moving the camera around, yeah, but just to show you over here, yeah, here, and now the camera got stuck. Okay, it froze. So what I will do is every now and then, for whatever reason, the camera freezes, and I don't know why. And this is kind of a little bit strange. Yeah. And let me see if I can deactivate and activate it again. Yeah. Yeah. Here we go again. Yeah. So you can see that's basically over there, right? So essentially, yeah. And essentially, from concerning image quality, the difference is sometimes not that surprising. So a microscope that costs 10 times as much does not necessarily allow you to see 10 times more. So where is the difference really? And the difference, or one of the differences, is really that convenience of use is of course one thing. So for example, this does not have a mechanical stage here. Yeah, you only stage clips, and a couple of other features, like for example, my microscope has so-called DIC. Which allows you to do these beautiful things here as well, right? And that's of course not possible with the lower cost microscopes. So, and you see that, yeah, sometimes you get pretty spectacular, yeah, images as well. And also, of course, I have more objectives connected. Yeah, so I've got, yeah, all the phase contrast and all these things here, right? So what I'll be doing is I'll be going through a little bit through the slide box, and we'll be comparing a couple of slides here, okay? So what I would like to do, therefore, is I'm going to go through the thing very quickly again, the chat every couple of minutes, and I'm going to look for some questions, okay? I also have, somebody says here, I also have some issues centering my condenser on my old Swift binocular. There are no centering screws, and I can't seem to use the screws on the side to center. That's interesting. Every time I try, it misses the center. Well, some microscopes have two centering screws, which are in kind of diagonal, and then you have to try to center it. I don't know exactly what the issue is here. Okay. What makes a microscope expensive or not? That's an interesting question, and that's actually yeah, a little bit of a can be answered in different ways. Well, the thing that makes a microscope expensive is, well, let me start differently. There is this 80-20 rule, and it's also known as the law of diminishing returns, and that basically says is that a microscope that costs 20% of the of another microscope allows you to see 80%. So with fairly cheap microscopes you can see quite a lot, but if you just want to have those occasional additional features that you really want to have, it becomes very expensive very quickly. Yeah, so that's a little bit the law of diminishing returns. Generally speaking, of course, optical quality is one thing that plays a role. Of course, modularity is one thing that plays a role, but there is one aspect that is completely ignored very often, and it has nothing to do with the microscope itself. And that is the whole, in the IT world you call it the whole ecosystem. So basically, if you have a cheap microscope like the one that I have over here, which I bought over Amazon, right? And it comes ready out of the box. But if I essentially want to exchange now one of those objectives, is this possible? Well, I can take them out. Look, I can unscrew it and take it out. But I'm not able to buy any replacement objectives because they're simply not offered. Yeah. So that is the thing a little bit. Yeah. If for whatever reason the LED breaks, well, then I have to kind of find my own solution to fix it, right? So this means is that those cheaper microscopes, it's simply not worth to get them serviced, and there is no possibility for upgradability. So that's basically the thing. And for the more expensive microscopes, there is more flexibility concerning this, and that has nothing to do actually with the microscope itself. So for example, if you're at a university or in research, they will always in most cases, fairly expensive brand microscopes, because if you have a problem with those, you get all of the replacement parts all separately available. For example, in our school, I'm a teacher, we wanted to once buy several microscopes, but we did not need the objectives because we wanted to reuse those, just the microscope body, and this was not even available. They didn't sell them because the microscopes come finished from the factory, boxed and everything, and there was simply no flexibility. And for the more expensive microscope companies, they will provide you this flexibility. Yeah? So the short answer is, what makes it a microscope expensive or not? Yeah, my answer is it sometimes does not have to do anything with a microscope at all, right? Yeah. So that's a little bit the interesting thing here. Yeah? So, yeah, horseradish to nematodes. Okay, you know what? I'll do that. I don't have horseradish right now, but because today's topic is a little bit a different one. Okay. Do you have any good suggestions for finding microorganisms now in winter time here in Northern Europe? Of course. What you do is you go out into the forest. You will see that yeah, there is still moss growing somewhere on tree stumps. You take a couple of these home, and what you do is you add a little bit of water and you give it a few days. Microorganisms will start to grow, and as a matter of fact, it's one of the videos, not a live stream, one of the videos that I made some time ago. Actually is I talked about finding microbes in wintertime. Yeah. So this one over here, again, you know what? I'm gonna go for the Loma. I have to find it again. This was also one of the things that I wanted to show you. I just know where it is. Here it is. I'm gonna make a separate video of this. This is a scary thing here. It's the stinger of a wasp. Okay. Now again, using my low-cost microscope, it's a little bit dirty and dusty, the whole thing. But a couple of years ago, I dissected a wasp. I didn't kill it. I found a dead one, and I said, of course, I'm going to directly put it under the microscope. So I made some, took some pictures using my stereo microscope, but then I dissected it again as well. And this is the stinger. So essentially, what you have is you zoom in, and we can do that maybe on the other microscope a bit better. Where is the arrow? I have to here. Let's try to make the arrow visible. I don't see the arrow. Just a second. It's a little bit strange. Here it is. Oh, here it is. I missed it. Right, there is a little canal in there for the poison, and there is some kind of a gland or over there which it produces the poison, and the poison is kind of directed through the small tube all the way to the tip. Yeah. So that is essentially a specimen that I prepared some time ago, and yeah, let's put it under the microscope, my other microscope, and let's have a look. Yeah, how it looks like there. It is a little bit dirty and dusty. I just realized. I'm looking for my microfiber cloth. Here it is. To do a little bit of cleaning. Okay. So this is the first one, was the tick. Now let's have a look at the stinger of the wasp. And I need to change around. I always have to switch cameras. Here it is. Okay. Focus a little bit. Yeah, let's comparable should be bright field, of course. Let's go down a little bit with the magnification as well. I have to flip out the condenser. Otherwise, where is this? Here it is. Here we go. Yes, here we go. You see, it's also the field of view is a little bit larger, and this is how it looks like. Yeah, with my slightly more expensive, better microscope. Yeah. I also have to say that, of course, the camera is better. Yeah, that I have here. Yeah. But yeah, concerning clarity, I think it's yeah. It's a bit better, but it's not such a huge difference. Yeah? But here we are now able to see also. It's not only a question of resolution really, but it's also a question of contrast. But look at this here. You're now able to also see that there's this thin tube in here, which is the duct for the poison. Yeah, then it kind of goes out here. I mean, honestly, it's pretty scary how I think at least how pointed, yeah, the stinger is. Yeah, it's a little bit dark again, so let's turn it up. Yeah, so this is a little bit another sample that I wanted to show you last week, but which I couldn't find in all of my mess on the table. Yeah? So I'm reading this again. Yeah. Do you have a reliable and a reproducible way to extract tardigrades from moss samples? Micropolo is asking, and the short answer is yes. But what is more important is that you have the right moss sample. The extraction itself is not the problem. And I found usually the way that if it takes me longer than a couple of minutes, 2 or 3 minutes to find a tardigrade, then chances are pretty good I won't find any at all, right? Go for different moss samples. So what you do is you take a moss, you add a little bit of water, you give it a couple of days, 3 days, maybe, for whatever living things are in there, including tardigrades, to absorb the water, come back to life. And then what you do is you rinse the moss, and you're going to see that a lot of sand and debris will be flushed out, including tardigrades, and then you look at that. Yeah? If you do not find any immediately or after a couple of minutes, chances are pretty good that the moss was not suitable. I found moss samples that are growing on exposed surfaces, on rocks, on I don't know, garden fences and so on, not so suitable. I found most tardigrades in moss that I've gotten from the forest, where there was no direct sunlight exposure. And I think I read somewhere in a research paper that tardigrades, while they're quite resistant to dryness, they don't always like heat very much. And I can imagine that if there's a lot of sunlight and a lot of heat, maybe that this actually might be harmful for them. So I found most tardigrades, and sometimes a lot of them, in moss samples, even dry moss samples, after I've added some water, which I got from a forest and not from those occasionally exposed surfaces. Yeah? So that's simply, and then what you do is you rinse the moss and you put under the microscope whatever you start to flush out. Yeah. I have the binocular version of the Swift microscope. I assume it has the same fold-out filter holder. Do the do-it-yourself filters showed in the previous video fit with that kind of holder? You know what, I'm going to switch over again a little bit here. The question was if you have a whatever microscope with this swing-out filter holder, yeah? And if you make essentially filters that fit in here, do they work? And the answer is yes, but you have to experiment, right? Because not all microscopes have the same diameter. So if you want to try, for example, dark field, then make several dark field filters with different diameters and put them in here, and then you choose the one that works best. Yeah, but generally, I've got several microscopes, all microscopes with a condenser, that's important, and a filter holder work. So in other words, those microscopes with which don't have one, like the one that I have on top here, yeah, you cannot do that. Okay. So what I'm gonna do now is I also want to show you something else. What I have here. This is a sand sample. So this is a slide which has indentations, so concave slides. And I put some sand in here, and the blue thing that you see over here, that is nail polish. So I used the nail polish as a glue kind of to kind of hold the sand in there. So this is essentially a so-called a dry mount. If you've watched some of my previous videos, then you've seen that I've also made slides using a ring, using a slide ringing table. But now let's have a look at the sand sample over here, using the microscope on top. And let's do this again here. And here it is. And of course, you always got to be aware that the camera readjusts its exposure. Okay, so that is also a thing that you have to be aware of. Yeah, and of course, as we some blue thing that you see on the side here, that's nail polish. You see that the sand grains themselves are kind of totally dark, right? Yeah. So what I recommend in this case that you do is to try something else. So we're gonna, so you see it kind of looks very washed out, yeah, and that is also because I don't know if the light here, it looks a bit. Also has to do with something a little bit with the camera exposure. But what you might also want to do is you might want to shine a flashlight on the surface here. How do I even turn this on? Okay. And when you do that, of course, you see that it looks a little bit more like a stereo microscope. Okay. But yeah, essentially that's also a possibility. This is the reason why some of the cheap microscopes, they also have a lamp from the top. I don't think that this is very effective all the time, right? But yeah, this is how it looks like here. Yeah. You might be able to see a little bit of especially when you have specimens with a very high contrast, you might actually see a little bit chromatic aberration. You see that there are some kind of slight color fringes here where the dark and the bright areas meet. Yeah, it's a little bit fuzzy, slightly bluish. Okay, yeah, so that is essentially chromatic aberration. Yeah. Nothing unusual. I mean, that is to be expected. But better microscopes have less of this chromatic aberration. Not the microscopes. Let's put it this way. Better optics, right? So this is the stinger again. So let's move this away, and let's compare this now with this one over here. So this is one of the and this you see essentially when it's dark, it's dark. Okay, so there is nothing but you might actually see not much better that the edges and the corners are much sharper and clear. Again, I'm having some issues with the arrow. Here it is. I always have to switch it on, right? So you see the edges now are a little bit more better defined. So let's move up with the magnification here. Even here, the higher magnification, you see that the corners are a little bit better defined. So what my general suggestion is if you want to do some kind of an optics test, get yourself specimens that have a very high contrast, sharp corners, and see how sharp those corners are. Let's have a look now as well. So I'm going to switch off the light here, and I'm going to use a flashlight now, also on my other microscope. This is basically how it looks like. Maybe I have to focus the light a little bit. You get the idea. It's also because of a different camera system. It doesn't look quite as washed out. Also because the camera adapter is probably a little bit better. So that is, where are we? Oh, that's the desk. That's the microscope view from the top again. So I'm going to read again a few questions or comments, if there are any. Why aren't all objectives replaceable? What is different between the different objectives? That's an interesting one again. Well, first of all, generally objectives are, now you have to look at the top again, okay? Objectives theoretically are replaceable because they're screwed in, right? However, you might not get the objectives sold separately. That's a little bit the issue. Why would you exchange them anyway? Well, because especially if your beginners are using a microscope, for example, in schools, very common, those microscope objectives do become damaged because some people, beginners, I mean, this can happen. Not even beginners, it can happen to more experienced people as well. You sometimes might actually crash up here. You know what? I'm going to switch over. You might actually sometimes crash the objective into the slide. Look, for example, this one over here does even have a spring-loaded front part here. So, and sometimes there are also some spills, and then maybe the lens becomes covered with, unfortunately, mounting medium. Also has happened. Some students essentially rotated the objective into mounting medium, and very difficult to clean. So of course it would make sense sometimes to have them replaceable. However, if they're not available, then what are you going to do? Okay, so that's a little bit the thing. And with more expensive microscopes, those are all available for different purposes. You have different objectives and so on for every type of application. But some of the objectives can be extremely expensive. And you don't buy them over Amazon, but you have to contact the company directly. And some of the objectives from the more expensive microscopes actually, when they become damaged, they're so expensive that you have to actually get them repaired. They will take it apart, the objective, and it will actually do the cleaning. So that's a little bit the thing that as well. So I'm just looking a little bit here again. This tick reminds me of how decades of and centuries of fine. So basically, tick populations exploding. Of fire suppression. Okay. Interesting. So I'm scrolling through all of the comments here. What 5 megapixel camera is that you recommend the brand? Well, I mean, let's put it this way. The camera that I have here on top, I have to show you. How am I going to do that? Or maybe I should, I don't know. Let me talk a little bit about the camera here as well. Okay, so that's the camera that I have. This is a 5 megapixel camera, and many of those microscope cameras, even if they are sold under different brand names, essentially the electronics is very similar. So this is a so-called from Tube Tech. That's the brand or the company that makes it the camera electronics, and then it's sold using different brand names. So it's a 5 megapixel one. I don't even know if it's sold anymore. Okay, but yeah, that's how it looks like. Essentially, I tell you what the bigger issue is with those cameras. It's not that is that usually the USB connection is a USB 2.0, right? And that is too slow for proper video. That's a little bit unfortunate because you do not get the frame rate. So those cameras are fine for taking pictures, but are too slow for video. Okay. So just maybe one or two more, and then I'll go through a couple of other. Yeah. When adding water to a sample, is it okay to add tap water if it is chlorinated? Well, it depends. If you want to, well, let's put it this way. If you want to observe water microorganisms, they are very sensitive to chlorinated water. But generally, when you have a jar with pond water, then you're just going to use that water, obviously. If you now have other samples like cross sections of I don't know, of plant tissues, of I don't know, whatever, then it doesn't really matter if you add chlorinated water, right? Because it doesn't matter if the specimen dies because maybe it's already dead anyway. So it depends really what you use it for. Water microorganisms are quite sensitive to chlorinated water, and you should not be using it. You probably don't need it because you have a jar with pond water anyway, and generally for the other samples it doesn't matter so much. Yeah. Can you explain again what is chromatic aberration? Okay, so basically chromatic aberration, I don't know if this is a good, I don't know if you have a good sample of this. Yeah. I'm just going to choose, just for the sake of it, I'm going to choose a different one here. Yeah. So that's a maze cross section. Okay, let's go up a little bit with the magnet. Is when an image that should be a white does not appear to be white. I don't know if this is the best sample either. Okay. Generally, it is like this, that when white light passes through an optical system, lenses, prisms, and so on, then the different colors of the light are refracted differently. And this can cause white light to be separated into its rainbow colors. And you can see this separation usually as the edges. Okay, yeah. When there is an edge between bright and dark, sometimes you will see some color fringes, like it's a little bit more purple on one side and yellowish on the other side. In this sample you don't see that so much, but usually this can also be seen of the image being a little bit fuzzy. And when you have a better microscope optics, more expensive ones, then essentially you see less of that and the image becomes a little bit sharper. Having said that, I would say the biggest factor that reduces image quality is not necessarily the microscope optics, but simply a badly prepared specimen, or simply not doing some color correction afterwards or contrast correction. You'll be surprised what you're able to, how much you're able to improve image quality simply by doing a little bit of image processing, right? So, but this here is, that's a cross section of maize, corn, the stem. You look at this again. Some people emailed me and they said they want to know a little bit more about the biology, right? And occasionally I've done that. But what you see, they look like little tiny faces. These are so-called the vascular bundles. This basically means this is where water is transported up the stem, right? And all of those little elements that you see here, these are of course individual cells, right? And those large cells here, they transport water up the stem, and they transport also sugar, sucrose, down to the roots, right? And now if you look at this, now this is now using the 4 times, then let's have a look at the 10 times. Okay, the camera always takes a little bit of time to adjust, and I think that those microscopes are, those cheap microscopes are surprisingly good, really. Okay, I mean, this is seriously, this is Yeah, it's kind of surprising what you're able to get already for fairly low price. Yeah? And usually the problem of image quality starts to become visible at a higher 40 times. But even here, and notice when I'm focusing, it's not quite smooth. It's kind of a little bit, the image is jumping. That has to do something with a slow camera transmission. You know, the frame rate of the camera is not quite high. Yeah. So, but now let's remember this a little bit, this image a little bit, and then let's put it over to the to the other microscope. So scope. So I'm going to now take this out again. Let's put it in at the top on my other microscope. I have to find it. Here it is. You see, one thing that you already noticed is that the field of a view is different, so you're actually able to see much more of the specimen at a given magnification. That has to do with the adapter optics and with the camera sensor size and so on. Okay, but let's zoom in a little bit. I'm deliberately not using DIC right now. I'm using deliberately the regular bright field. Yeah. So my the camera is not still pointing to the other microscope, right? Yeah. Yeah. And this is how it looks like. Yeah. Let's go up yet further. I have to adjust a little bit. So, and you see that this is how it looks like using the other microscopes that I have. Yeah. You do see that the image does look crispier, and because this microscope has also a condenser, I'm also able to control contrast and depth of field this way. Yeah, yeah. The light intensity goes down, of course, when I close the condenser, but yeah. So I can make certain adjustments to this here as well. Okay. I meant why microscope objectives in different companies, like why can't we replace a Swift one with an, let's say, Olympus objective? Oh, this is an interesting question, okay? So the question is really, if you have a microscope of a certain company, is it possible to use objectives of a different company? And the answer is you're gonna hate it. It depends. You see, generally with modern microscopes, you cannot exchange them because the modern microscopes have so-called infinity corrected optics, and they're not compatible. So if you've got an Olympus microscope, you have to use Olympus objectives. If you've got a Nikon, for Nikon, Leica, Zeiss, it's all company specific. But having said that, those companies, they have a large selection of objectives anyway to choose from, and they can be quite expensive, right? Now for those introductory microscopes like you see up there, these are non-standard objectives. These are tiny objectives, and I couldn't even find replacement objectives for those. Probably because it's not worth it, because the microscope itself is already so cheap. And then there are the other microscope objectives. They have the number 160 written on them. And those are generally, with some exceptions, exchangeable among each other. But what you should do is you should not take out one objective and then insert another one of a different series, because you're going to lose focus from one when you change from one objective to another objective. You see what I mean? It's all complicated. Yeah. So generally, it is possible in some cases to exchange objectives, but you gotta know what you're doing, right? So I've once got myself a new objective. I put it in, not into this one, but into another microscope of mine. Did it work? Yes, it worked, but I had to refocus every time. So it was not quite as convenient to use. Yeah. So, but generally, between companies, between those big manufacturers, you cannot exchange objectives. Yeah. What water do we use for pond water then? Well, pond water. I'll give you an example here. Look, I mean, I've showed this to you already. It's pretty green. So this is contains, I don't know, lots of ostracods, algae, and so on. This is just plain pond water. Yeah. So if I want to take another sample from here, I'm just going to use that. Yeah. There is no need to mix it with tap water. So this is a little bit the thing. And if you have, for example, if you want to get some tardigrades out of moss, and you want to use tap water and it's chlorinated, then don't. get yourself some bottled water or some mineral water and allow the carbon dioxide to escape, or use tap water and let it stand open for a couple of days for the chlorine to escape. But tap water does have another problem sometimes. Depends on how sensitive they are. If you have a metal tubing in your house, then if the metal content of the water is too high, especially if there's certain metals in there that shouldn't be in there, like lead, for example. Why should there be lead in the water? Well, because in some very old buildings in the long time ago, in some countries they used lead. They had lead in the pipes, and this kind of is not good for the microorganisms either, right? And also not good for drinking. And I don't even know if it's allowed these days. Those cells look really washed out. I wonder why. Maybe it's because of the manufacturing process. You mean those ones that I show you right now over here? Let's put it this way. Maybe it was washed out. You notice that the cells here in the center, they look empty. Where are all the cell organelles? Where's the nucleus with the DNA, the mitochondria, all of the things that a cell normally has? It's not in there. The only thing that you see over here is this thing on the side here, and that is the cell wall. The reason why it's empty is because these are very, very thin cross sections, and the cells are actually cut open, and all of the cell contents were lost during the preparation process, but this was done deliberately. Yeah? Because if all of the cell contents were still there, then this whole thing would look a little bit more messy, so to say. But the way that they made it is they wanted to have this nice pattern-like appearance that we have over here, right? There's a little bit of dust there, but the cells, they should be kind of empty. Otherwise, it wouldn't look so, how do you say, so clean, right? So it really depends on what you want to do. Yeah. So this kind of, maybe this was kind of the question. Yeah. So is the objective thread on the cheap microscope not RMS? Correct, it is not RMS. RMS threads, RMS stands for Royal Microscopical Society. And those here are non-standard objectives. They're significantly smaller. Okay? Yeah. So that's the thing. These are not RMS ones, and therefore, yeah, you've got problems finding replacement ones. Do you have also a centrifuge device for more dense concentration of particles? Yes, I have a centrifuge, and as a matter of fact, some time ago I did also make some live streams where I demonstrated the centrifuge. You have to go back a little bit in the live stream. Yeah. So okay, and there's another question here. Then I want to put some more samples under the microscope. I meant that when a pond water sample is dry, do we add pond water or just regular bottled water? And you also mentioned something about mineral water too. Well, yes. Well, the thing is the following. If you have a sample, I don't know, I can show you here, like this here. I mean, this is a pond water sample, right? Yes, I don't know if you're able to see this properly. Let's go to the desk here. Okay. You can see there's some solid material here, some algae floating around. And I've kept this sample open now for several months. And what I have done is every time when the water evaporated, I added a little bit of non-chlorinated tap water. And if you always add a little bit of water, then the osmotic stress on the microorganisms is not going to be too high. So for example, if you have a home aquarium and you also have to exchange the water, then you do not exchange the full water, you just exchange always part of it. Yeah, maybe 20% or 30% of the water, because then the stress on the fish is not going to be so high. If the whole sample completely dries out, then many of the microorganisms are going to die off. And then when you add water, it doesn't really matter which water you add, you're not going to get them back to life. Yeah, so I've actually tried that. Don't let the sample dry out completely, because you're killing off the microbes, and most of them. And then you don't have anything interesting, even if you add water and wait for some time. Yeah? So always refill a little bit of water, but not everything, right? And something about mineral water or bottled water. So if you have, it's better to use some bottled water and to fill it up than chlorinated water. Yeah. As a matter of fact, I've read somewhere if you want to grow certain algae, you should use bottled water, because some of the metallic ions that you find in tap water, depending if you have metal pipes, are not so good. So these are just some recommendations. So you know what, I'm going to go on and I'm going to work myself a little bit through the slide box here. Yeah, there are a couple of things. Okay, this one is a stress test for the microscope. Skeletal muscle. Okay, and what I'm going to do is I'm going to show you. I've done this already before, but I'm going to show you now some skeletal muscle using my high performance microscope. And because this is a real test of if how well the microscope is able to resolve this. No, that's the wrong one. Here, this one is the one here. That's muscle. Okay. I have to look and find some of the muscle fibers that you see over here. And let's go up a little bit with the magnification. So this is bright field, obviously. Okay. I'm just looking for something here. And let's go 40 times. Okay. And yes, and a little bit here. This is because it's so difficult to see. These are the so-called the striations, one of my favorite specimens here. Look, if you look very carefully, you might be able to see that there are some vertical stripes here. Okay, these are the striations because of the protein fibers, actin and myosin. They make the light and dark bands. So they are these are the muscle fibers that run horizontally, but they're those fine vertical striations that you see here, right? Now I'm able to see that with my high performance microscope, but let's see if we're able to see them also with a low cost microscope. Probably not. Okay, so that is essentially the thing, that I would like to show you. So that's 40 times. So we're going to be using the same magnification over there, but I can go up to 60 times as well. Yeah, now you see them much better. I'm going to open the condenser. See those stripes here? That's why it's called striated muscle. Or skeletal muscle. It has those striations here, the light and the dark bands. So let's see if we're able to see those using my low-cost microscope. Okay, so let's take this out again. Let's turn down the light a little bit. Let's switch over to the stereo microscope. No, not this one here. Here we go. Okay. So let's go up 10 times, 10x. Okay, you see the camera is trying to always adjust its, a little bit. And now 40 times, that is now the same magnification as with my other one. I have to carefully focus. Surprise, surprise. I underestimated the microscope. Look at this. Yeah, even with a low-cost microscope here, we're able to see those light and the dark bands. Okay, the stripes. Well, actually, I didn't expect that. Actually, it's better than I anticipated. Yeah, I just wanted to show this to you. Okay, would it be possible to connect a DSLR to the cheaper microscope, even if it's only monocular? The answer here is if you try to connect a DSLR to the microscope on top here, it's probably going to be a balance problem. Okay, that is the problem. DSLR cameras are so heavy. And what they're going to do, the cameras, the DSLR cameras, they're going to, because they're so heavy, they're going to impose quite a big load here on the tube, right? So you can connect pretty much anything, obviously, with the right adapters. The question is it worth it? I would generally recommend, before you connect the DSLR, where probably the whole adapter system costs more than the whole microscope, I'd probably get myself a cheap adapter for a mobile phone and connect that to the eyepiece, right? And then you also have like a little screen that you can use. Yeah, so I would probably say that this is more convenient. Yeah? Theoretically, it's possible to connect the DSLR, but whether you might need to add maybe a little tripod or some additional support. Yeah? So that's a little bit. Yeah? Oliver, why don't you still use the really cool music intro in your videos? Yes, that's a good one. There was if you watch one of my early videos in this channel back 2018, 2019, I had an intro, a couple of seconds in intro with a very nice music, and I video edited and looked pretty cool. And I bought that music, so I bought the license rights to be able to use those. And after some time, I decided not to do the intro anymore, even though I have to admit I liked it a lot myself. So if you don't know what I'm talking about, do visit my early videos. And the reason why I decided not to use the intro anymore is because, yeah, it's a little bit of a YouTube thing. People they click away. So people who essentially already know the YouTube channel, they feel a little bit tired maybe of the music, and some people will also tend to click away. So there is generally the recommendation these days to really jump to the point directly and not spend too much time on an intro. So it's a little bit more of a how do you say a YouTube watch time, YouTube percentage view, video view issue. But I have to admit, I liked it myself. Yeah. Did you cut your chin shaving? This is the question here. What's this dot here? Yes. Yeah, I was. I cut myself today in the morning. Yeah. Someone in your other video commented, "Can you leave objectives not totally screwed in? What does it do?" Ooh, okay. So listen, again, look maybe on the top here. When the object, you know what, I'm going to demonstrate this. I'm just going to demonstrate this on this sample here. Let's say that it is not completely screwed in. This has actually the effect, look what happens. It defocuses, okay? So I can get it back into focus again. So it's not screwed in completely. Yeah, I can get it back into focus again. But look, it's very unstable. And the bigger problem is the following, that because it's a little bit out, it is now lower to the cover glass and to the slide than the other slides. So this basically means there is now a higher risk of actually crashing into the slide. Yeah? So generally, this is not something that you basically want to do. As a matter of fact, I once noticed, even on my other microscope, that when changing from one objective to the next, I lost focus. And I said, what happens to the microscope? Is something broken or so? And then I discovered that actually the objective was loose, right? And then, yeah, you don't get the best image quality. Yeah? So you also have to understand that even if you're able to get it into focus, then essentially you might not get the best in image quality, even if you refocus it, yeah? Because the distance is not correct. Yeah? So this is thing here. So yeah. Thank you for all your very informative videos. I recently purchased the Swift SW380T. And I'm just waiting for some slides and camera adapter to arrive. Yes. So basically, what I said at the beginning of this video about the centering, so that is it's exactly the condenser of the Swift microscope. Okay, so close it, and if it's not in the center, then make the screws. There are 3 screws here, and here. Loosen them a little bit, center it, and tighten it again. Yeah. So that is this is exactly the same condenser of the Swift SW380. Okay. I'm particularly interested in imaging fungi and mold, and also saltwater marine microorganisms. Any unique considerations for these types of samples? No, not specifically unique. It depends more on the specific type than from where you got it from. Saltwater marine microorganisms do have the following issue, that as soon as the water evaporates, the salt concentration rises. So just be aware of that, especially if you keep marine water in a jar like I have over here. Okay, this is approximately 2 years old. There's some sand on the bottom, marine water from northern Germany. Okay, the diatoms are still alive. I'm very happy. So there is quite some movement going on. And you notice the following. I put a lid on top to prevent evaporation, because if water evaporates, the salt concentration goes up, and this might not be good for the microbes. On the other hand, what you have to do is, if you put a lid on, you always have to make sure that there is enough air in here. Okay, so never make the mistake like I did some time ago. I took on holidays some of those tubes along, right? Fill it up all the way to the top with water, and then put a lid on top. And this immediately made the whole system go anaerobic. I put it into light so that there's photosynthesis and an oxygen formation. It wasn't enough. Always when you collect water samples on a holiday or whatever, make sure that you can do that. Yeah, and it will stay fine for several days, provided that there is enough air on top. So that's basically a consideration that I have. Concerning fungi, don't forget that spores, don't inhale the spores, right? And if you would like to observe some fungi, and then I would suggest take those as a starter that from food, not spoiled food, but from certain different types of cheeses, for example, and so on. So basically food sources. Yeah. So that is also mold. There are certain camembert cheese which contains mold. Yeah. So there is plenty there. Yeah? I'm not sure which microscope to buy. Do the following. If you are really not sure and you want to start the hobby, get yourself one of the very cheap one, not toy microscopes, get yourself a cheap one like this. I know it's limited, okay, but you have not lost anything concerning the money. Yeah, it's around €100, $100. It's not so expensive, and then you can always decide on whether you like the hobby or not. Yeah? And what else do I have here? Kieslein. Okay, yeah. So these are diatoms. So where are some diatoms? Let's see those. Let's have a look at those. Diatoms are also usually used for checking the resolution. Okay, and these seem to be round diatoms, are often marine diatoms. So let's have a look here. Let's get rid of the arrow again. Okay, yeah. So this is now the 10 times objective. So these are diatom shells. Diatoms, of course, are related to the algae. They do photosynthesis, and these are just the silica shells of some of the diatoms. Very often marine diatoms are round ones, and these are just fragments here. Let's focus on this guy here, and let's go up to 40 times. Here we go. You see, that is a little bit one of the difficulties now, is how do I center now this slide without actually, if I bump into it, so here we go. So these are diatom shell fragments. Okay, using again my low-cost microscope, 40 times magnification. Yeah, total magnification is of course higher, a little bit. So, and now let's do the following. I'm going to now show you how this actually looks like now with my other microscope. Okay, so I have to switch over the scope view. Here we go. Okay, I go down with the magnification again because I have to find something interesting here. I don't know, maybe this one over here. They are randomly scattered around. Back in the 19th century, what some people liked to do is they try to actually arrange them nicely in patterns. So they have made microscope slides with nicely arranged diatom patterns. How do you arrange them? What they've done is they have taken their own eyelashes and mounted them to a little stick and actually moved and picked up diatoms this way. Because the eyelashes are a little bit greasy and oily, and so the diatom shells were actually sticking quite well, right? So, and this is now how it looks like with the other microscope. Okay. Yeah. You see that it's not entirely flat. That's why when I focus through it, then some parts move in and out of focus as well. Yeah. So I'm going to read again a couple of questions here. With collecting in tubes to maintain oxygen level for a few days, would a 50-50 mix of sample and air be sufficient? Or would it be better to go with 25 sample, 75 airspace? Please go with 25% sample and 75% airspace. I will tell you why. Back in the day, I mean, that's an empty one, almost empty. What I've done is I collected on a holiday a couple of those tubes and I not thinking. Yeah, said I want to take along as much sample as possible, completely fill it up, right, and put a screw cap on it, and then when after 3-4 days, back home I opened it and it smelled bad, sulfur. Oh, it's gone anaerobic. And this kills off, of course, many. And then I had one tube, which I only had filled in very little, right? And everything else was essentially air, also screwed tightly, absolutely no problem. Okay. So my suggestion is give it a little bit of more air, and then every day or so you can always open it up and yeah, exchange it. And my suggestion is if it contains green algae or anything green, then it does photosynthesis, and this so keep it in a light area, bright area, but not in a hot area, because if it's hot, then the sample is going to heat up, because you have a little glasshouse effect, greenhouse effect in here. And it's going to heat up, energy is going to go in, it's going to heat up too much, and it's going to again destroy the sample. So keep it in a bright place so that the algae are able to do photosynthesis, but not in direct sunlight. Yeah. So this is something I can recommend. Yeah. So looks like the sunflower of the central part. Okay, no, this is a diatom, but if you already mentioned the sunflower, then I'm going to put a sunflower in here as well, because sunflowers also look nice. Okay, these are made of silica shell. It's of a diatom, but a sunflower is that I like to show you this because it's again one of my favorite slides here. Okay, looks like this. See, it's a little bit too bright. Yeah. So this is basically how the sunflower looks like. Yeah. The vascular bundles are arranged on the side, so those cells transport water up the stem, and that's how it's different from to do a little bit of botany, this is a so-called a dicot plant, which contains the vascular bundles on the side, and the maize, the corn stem, which I just showed you several minutes ago, actually is a monocot plant, which has all of those vascular bundles scattered throughout the stem. Okay. So let's remember a little bit how this looks like. Let's go all the way to 40 times again. I have to always, yeah. You can actually see that some of the cell walls are fairly thick. Okay. It's quite a, yeah, I think quite a nice specimen. It's professionally made. It's commercially, it's a commercial slide. Okay, so now let's put it under the other microscope as well. And where is it? This one over here. Okay, let's start again at the low magnification. Okay. Looks a little bit, the resolution is not so good with a 4 times objective. It's kind of interesting. I have to also say that it does look a little bit better with when looking through it directly and not with a camera, because I do think that the camera also adds quite a bit of additional magnification to it. And then let's have this one over here. Yep, that's now the 10 times objective. It looks pretty okay, I would say. It's fairly okay, not quite as crisp as the other one, but it's, I'm always surprised about what's possible with such cheap, relatively cheap microscopes. You can do some serious hobby microscopy also with the cheap ones, even if they're not quite convenient to use, because I have to move it around with my fingers and all these things, you know. Let me quickly, readjust the camera here. Yeah, and now 40 times. Yeah, that's how it looks like here. Again, here I'm not able to make any adjustments with a condenser because I don't have a condenser here, right? Yeah. Of course, also moving it, the slide with fingers is not quite as easy. Yeah, as one might think, but it's still possible. Yeah. Do monocots, biology question, do monocots and dicots have their own advantages? Like, does the position of the vascular bundles affect anything? Let's put it this way. I'm getting questions like this fairly often. What is the advantage of one system versus the other system? And maybe we should answer the question differently. It doesn't, both monocots and dicots, it works for them. So you can, if one system were so much more advantageous than the other one, then the one would not, the other one wouldn't exist anymore. So usually in nature, if you have multiple ways of actually solving a problem. And for monocots and dicots, these are just two different plants which have two different arrangements of the vascular bundles, and it works for each one of them in their own way. Yeah. I mean, it's a little bit, I know this analogy is a little bit, let's put it this way, it's a little bit like asking what's better, a car or a bicycle? What's better? You cannot say that. I mean, each one of them works, works, you know, and that's why we have both, right? So that is a little bit the thing here, that sometimes you cannot always necessarily see advantages or disadvantages. It simply works, and therefore nature kept it. Yeah, it has not been, it didn't die out because it didn't work, right? So that's a little bit the answer here. So I wouldn't be able to say that one, is necessarily better than the other one. Yeah. I have a cheap microscope, and only now, when doing some imaging, I noticed that there is a little blurry from 100 times magnification onwards. Is the cheap objective or the slide to blame? I would say, well, what do you mean from 100 times onwards? Generally, if you have there's the cheap objective or the slide to blame. Generally, it is like this, that microscope, doesn't matter if it's a cheap one or not a cheap one, the larger the magnification that you have, the blurrier it becomes. Yeah? So that is that's generally the thing. So subjectively, an image will always look more crisp at a lower magnification. I just want to show this to you here. Again, over here. Yeah. And this has nothing necessarily, it might be also with a lower depth of field. Look, I mean, this kind of looks very sharp and crisp, but when I go up to a higher magnification, okay, it looks more fuzzy. It's not quite as crisp, okay? And the reason is, or one reason is that at a higher magnification, you also have a lower depth of field, so you have a more of an overlap between those parts that are in focus and some parts are out of focus, and this makes it appear fuzzier. Yeah. So what I'm saying here, it doesn't this blurriness doesn't necessarily have anything to do that the objectives alone are not good. Could be, but I would say there could be other reasons that have a bigger impact. Yeah. It could also, of course, be that that the specimen is fairly thick. In that sense, the slide is to blame a little bit, right? Because if the specimen is very thick, then some parts will be out of focus and will therefore appear blurry. So subjectively, it will appear to be more blurry. See, now other parts are in focus. Yeah. And if I go back to a lower magnification again, then because the depth of field is so much larger, yeah, then everything will appear to be in focus, and everything subjectively appears to be more crisp. Also because you don't see all of the details as much, right? Yeah. So yeah, that's the thing here. Yeah. Eyelashes, incredible. So basically, I think this refers back again to the diatoms, and indeed, what they have done is to how do you make diatom arrangements on a slide. Indeed, they've taken eyelashes, and if you put an eyelash under the microscope, you're actually going to see that it goes together to a very sharp point. But at the same time, the eyelashes are flexible and they don't damage the object. And because they're greasy, they're sticky, and therefore you're also able to pick up the objects, right? I've never tried this myself before. I can only say that what I've read in a book. Yeah. What is depth of field exactly? We can actually, I don't know if I'm able to do that. I don't have a pen now. It is basically depth of field is the part how much of the sample from the top to the bottom is in focus. That's the depth of field. And essentially, if you have a specimen that is, I don't know, maybe I can show you over here. Yeah. Let's go to a very high magnification again. Yeah, you see, yeah, look, have a look at this line over here, right? Yeah? This line is clearly defined, is in focus, but the one over here is out of focus. It's fuzzy. And now look what happens when I turn the fine focus knob. I have to turn it into wrong direction. See, it's in focus. And now I turn the focus knob, and now the other line over here is in focus. Yeah. The reason is because they are not in the same plane. Yeah. So they are, yeah, and how much at the same time from the top to the bottom is in focus, this is referred to as the depth of field. And notice when I go now down to a lower magnification, again it might be a little bit more difficult to see because yeah, at a lower magnification, yeah, what you're going to see more easily is that essentially both the top and the bottom part are both in focus because the depth of field is larger. Right, so you're actually able to cover more of the, yeah, and the question now is, that's an interesting one, is it better to have a larger or a lower depth of field? And the answer is have you guessed, it depends what you want to do, right? So what I can do now is I can open the condenser. I have to go down with the light intensity, and when I open the condenser, what will happen is that the depth of field will go down. Yeah, so I can now more parts will appear to be fuzzy, right? But this one here is in focus again, and the thing is if some parts are fuzzy, some people say that's not good because it looks blurry. But I say in some cases it might be an advantage because the fuzzy parts then are not able to cover up those parts that I would like to look at. Yeah. If everything were in focus, then sometimes I'm not able to see very clearly the part that I want to see because it overlaps with another part that is in focus, which I'm not interested in, right? So this is a little bit see, all of a sudden I closed the condenser now. Now I can see all of the dust and the dirt and some other stuff that I'm not so interested in. Yeah. So so this kind of disturbs the image a little bit. Yeah. I was lucky to get a used PowerCo BMT series microscope 40 times, okay, with the mechanical stage of eBay for $35 US. Wow. I don't know the brand, honestly. Okay. And indeed there are quite a lot of microscope companies out there. And essentially, I always say it really doesn't matter which microscope you have or which brand you have, as long as you enjoy using it. The best microscope is always the one that you end up using the most often. Yeah. So let's, I don't know, I'm just going through a couple of other slides here. What time is it? Oh, it's 1 hour 12 already. 1 hour and 12 minutes. Yeah, maybe you want to look at this here. It's kind of a fun one. It's not an easy specimen, as a matter of fact. This is, it's very dusty and dirty. This is the cross-section of a fly head. Okay. And I just want to show you quickly also something here. It's not, it's the slide itself is not very. So this, let's leave it like this here a little bit, okay? Just wanted to, you're going to see the image quality is not very good right now. It's a little bit overexposed as well. And because they have the camera, but at the same time you see that there are these, I don't know, do you see this kind of ring over here like a reflection, right? Yeah. So this is an extreme case of stray light as well. So better microscopes, they also have more expensive microscopes. They also have so-called Köhler illumination, which allows you to improve the contrast and to prevent things like this from happening. Yeah. But you can compensate this by actually doing a little bit of image color correction. Okay. Yeah. But then again, the camera itself also tries to adjust the exposure. So let's go to a higher magnification now. Yeah. Yeah. And so this is now using a 10 times. Yeah. Yeah. There is, I don't know if I'm able to shield this off somehow a little bit, yeah, but there is still some internal reflection here. And often when the specimen is quite dark, like for example the head of the fly over here, then of course you're able to see those internal reflection which are brighter much better, right? So this is just something I wanted to. This is a difficult specimen also for another reason, because it has such a high dynamic range. What do I want to say with that? Yeah, just want to show you. This here is now the tip of the trunk of the fly. Okay, now properly exposed, but it is actually much brighter than the rest of the head. Yeah, so I'm going to show this to you now, also under the other microscope. Yeah, so scope is over here. Okay, so here it is. Okay, I'm going to flip out the condenser again. Yeah, so I just want to show you the it's again, I should clean the slide a little bit. Actually, what I wanted to show you is that over here right now, this part is quite dark. You're not able to see a lot of details. Huh? And the trunk down here of the fly head is relatively bright or correctly exposed. But so if I were to make the whole thing brighter, then I'm able to see this part correctly exposed, but the bright parts are too bright. I'm just saying this because some time ago I struggled quite a little bit with that because I couldn't get a properly exposed image here. And if you have a problem with dynamic range, as it is called, which is the difference between the bright and the dark parts, then you have to do multiple pictures of the same specimen and then combine it on the computer to get both parts properly exposed. Yeah? But I just also wanted to show that this is also something that that better camera systems are able to deal with, like DSLR cameras like I have, is they're better able to capture details in the bright and also in the dark areas. Yeah? And especially insects which have such a dark exoskeleton, for them it's particularly a problem, right? Yeah. So let's zoom in a little bit more. It's of course very nice to have a mechanical stage here. Yeah. It's fairly bright already, and over here, yeah, almost no details visible, right? So I have to go up with the light intensity, yeah, like this here. Yeah, now I'm able to see more details, but this actually means, like you see over here, the image appears to be blown out, yeah, and overexposed. Yeah, so I'm losing details over here, yeah, and but I see more details over here. So you cannot have it both. Yeah? And this is actually more of a camera problem. Yeah? So generally, I'm now using a DSLR camera, and generally DSLR cameras or large sensor cameras are able to deal with a dynamic range problem better than cameras with a smaller sensor or cheaper cameras. Yeah? So I just wanted to make this clear to you that image quality does not only depend on the optics, but also on the camera system used. And I wanted to show you this here as a rather extreme example. You see, you almost don't see any details anymore, right? And if you go down with the light intensity, yeah, and now you're able to see it properly, yeah, and now you're happy with that. But then, yeah, at the same time, it's too dark up here. Yeah. So that's a little bit the trade-off that we're having here. Yeah. So the thing is, I don't think we can see any cells here, right? And as a matter of fact, you are right for two reasons. First of all, this has been prepared in such a way that you see the exoskeleton, which is made of chitin. So maybe there's still some residual cells somewhere. But you have to understand the following. Flies are, believe it or not, animals. Okay? And animals do not have a cell wall, and therefore it's almost impossible sometimes to see where one cell starts and the other one stops. And I'm going to show it to you on another example. Okay? Just because you wanted to know about the cells, this is now from a human scalp, so from the head, right? Look at this, okay? All of those this red thing that you see over here, that is a cross-section of hair. Yeah? And if you look just carefully here, it's also kind of difficult to see where does one cell start and the other one stop. Sometimes you are able to see it over here. These seem to be like fat cells, adipose cells, but here it's kind of already becoming very difficult. And these are not cells, but these are individual muscle fibers in cross-section. Yeah? And the reason why in animals we're not able to see the individual cells sometimes very easily is because they're joined at the cell membrane, so two cell membranes touch, and the cell membrane is so thin that you're not able to see them as different. With plants, it's different. They have a huge, thick cell wall outside of the cell membrane, so it's much easier to see where one cell stops and the other one starts. Maybe this is also the reason why they discovered first that all plants are made of cells, and it took them some time until to discover that animals are also made of cells. So let's go back again over here to the to the cheap microscope. Here, this is how it looks here. Okay, so still I think fairly okay. Yeah, I'm always surprised what those low-cost microscopes are able to do. Yeah. So if you have one of those cheaper microscopes, then you can do some interesting hobby microscopy as well. But again, you see, it's not quite as convenient to move the slide around, right? Yeah. And let's go up again with the magnification. Here we go. Again, it's a little bit slow. The camera response is a little bit slow. Yeah. Yeah. So let's go through. Oh, okay. I'm going to do now the following. I'm going to go through a couple of remaining questions, and then I'm going to think. I think I'm going to call it a day. Or are the cells just too small? No. Well, they can be small, but no, they're not. The microscopes generally have no problems resolving cells. Yeah, eukaryotic cells. Yeah. Wonder why the condenser, like infinity objectives, are not part of new microscopes in our modern times. Why the condenser are not part of new microscopes? Honestly, condensers like the ones over here are actually part of a regular microscope. Only those introductory microscopes, like I have this one over here, doesn't have one. Why? Because they were saving a little bit, right? And a condenser, as you can see, is not absolutely necessary to get a picture. I think it's for anything which is going. If you want to have it a little bit more advanced, you should have a condenser. But you see, you are able to get a picture even without one. Yeah? So it is really a question of cost, right? Do ordinary microscopes have almost similar numerical aperture objective lenses? Let's put it this way. Yes. If you get similar, not always the same. If you have, let's say a micro, like the one that I have over here, a 10 times. If you have a 10 times a microscope objective, it has a numerical aperture. This one of 0.30, for example. Yeah, a different manufacturer might have maybe 0.25 or something like that, right? But I would say that the numerical aperture alone is not the only thing that determines image quality. It also depends if it's a plan objective, for example, and so on. Yeah? But generally, the numerical aperture is something that determines how much is what's the angle of the light which is able to which the microscope is able to capture. The microscope objective is able to capture, right? So it's also something of a geometric measure. Yeah. So can you explain what are infinity objectives? And infinity objectives are found mostly on more expensive and better microscopes. Infinity objectives themselves do not have any significant advantages or disadvantages, so they're not automatically better, right? But essentially, infinity objectives are simply the way that I don't want to get too technical about it. It depends a little bit on how the image is created. I'm just going to use this here as an example. This is not an infinity objective. Yeah, basically it works like this. Let me see this here, right? Microscopes objectives is what they do is they work like a lens. They create an image over here somewhere in space, and then you have an eyepiece and you look at this image, right? Yeah, so that's kind of like a lens, and it creates a so-called a real image over here in the image plane, which is then magnified by the eyepiece. And in infinity objectives, what happens is that the objective does not create directly an image, but it creates a parallel light beam. Then you need another lens, a so-called a tube lens in here, inside the microscope, which then creates an image. And because the light is parallel here, in other words, focused to infinity, you can change the distance here as you like. And this allows the microscope companies to make microscopes that are bigger in size. And you can also insert filters in here which do not change the focus. So there are a whole bunch, a few technical advantages for having infinity objectives, but they on their own do not automatically provide automatically a better image quality. Yeah, so it is more also something that gives the microscope more flexibility. Okay. 160 millimeter objectives are standards. This is important? Yes. Basically, this one over here is not even 160 millimeter objective, standard objective. It's just not even standard, because it's a fairly cheap microscope. Yeah. What do we have to consider purchasing a DSLR adapter? You have to consider the fact that you actually need a so-called a T2 adapter ring, which fits your camera. So usually these rings are easily available for Canon and for Nikon, but if you have any other DSLR, you have to make sure first that you're able to connect the microscope DSLR adapter also to your camera. That's a little bit, also if you've got a mirrorless camera, you need a T2 adapter ring for mirrorless and so on. So actually getting the adapter connected to the camera is also important, obviously, and you need a correct adapter ring. Yeah. Is there a way to increase depth of field if you don't have a condenser? Yes. I can actually show you, but it's highly experimental. And you're going to lose a lot of light. So if you have a microscope like this one over here, which does not have a condenser, I'm going to show you. This one here has a condenser. You can open and close it. But what you can do, and I cannot show this to you now because I cannot flip this around, or maybe I can, I don't know. Can I do this? Can I show this to you without this? Yes, I'm now, look, this is the microscope here on the bottom. Look at the top. Okay, look at the top. You see that there's this ring here that you can turn with holes in it, which absolutely are completely useless because the holes are way too big. Yeah, and there is a screw here. What you do is you remove the disc, by removing this, by unscrewing it here, and then what you do is you take a tin can, Coke can, whatever, right? And you cut out a little piece of metal and you glue it over the hole, okay? You glue it over one of those holes and you make the hole much smaller this way. So what you're essentially doing, those holes are so big that they're completely useless, right? But by actually where can I show this to you, right? Here there's this ring here that you can turn, right? With the holes, and by making it much smaller, what you're doing is you're essentially stepping down the condenser, and this will result in a significantly darker image, but a much larger depth of field and sometimes also more clarity. Now, the darker image, if you have problems seeing a picture, what you have to do is you simply have to increase the exposure time. But I tried this and I was actually able to improve the image quality of this microscope by actually making the hole smaller, right? Yeah. How can I stitch HD screenshots into a single? There are you can make a high HDR. There are programs available that do that. Some programs like Photoshop and so on are able to do this automatically at the click of a mouse button. there are different ways how you do that, and I would suggest that you watch your own separate YouTube tutorials on how to make HDR images. So what you do in this case would be, let's go back here again to the, okay, this would be actually also the topic of another video. What you do then is the following: you take pictures here, some of them, extremely bright, you take a picture, and then the darker, you take a picture, darker, you take a picture, darker. You take always, different exposures, from bright to dark, and then you have got your 5, 6, 7 different images, and then you put them into the program, and the program will generate one image, one HDR image, where all parts are properly exposed. So it will take the from the brighter ones, it will take this is generally overexposed, but then it will only take the correctly exposed ones from this here, right? It will ignore the rest, and so essentially the software will actually combine this. I've seen that there are a couple of online tutorials. There's several ways how you can do that. Does the quality does the quality of a condenser significantly affect the image quality, like comparing the condenser of a 400 microscope and a 2000 one? I have not done this comparison. Let's put it this way. I can imagine, like anything, is quality dependent. It can, but the question is whether the difference is large enough to be significant. Optical coatings might play a role. Internal reflections might play a role, obviously. But the question, I would say, is more also one if you're not, you see, with the best microscope, if the image is not properly processed afterwards, you don't have a good image quality, for other reasons. What's my most precious microscope? Well, I've got several of them, but I would say the one that was my first one, my first microscope, which I bought back in 1998. It's an Olympus CH40 microscope, which I bought right when I graduated university as a little present to myself. Was really, I saved for a very long time for that microscope, and for this reason it is I have a very high personal connection to that. It's not the one that I have right now over here. I've got it somewhere else. I've been trying to ask that question about the adapter since the beginning of the live stream. Good thing to know. YouTube blocks comments from nicknames that have bad words inside. Oh, I see. I see. Okay. Interesting. I didn't know that. Okay. No, because sometimes it happens like this, that there are comments appearing and I see them, the comments, and then later on the comments are all of a sudden they're gone. And I did not remove them. So, and I don't know what happened to them. Okay. What about the focus with a T2 ring? Don't worry. The T2 ring is designed in such a way that the focus is correct. And this basically means that for different cameras, the size of the T2 ring and the thickness is different. Okay, that's the whole point of the T2 ring. There are standards for this. And that, for example, for mirrorless cameras, which have the sensor much further in front, the T2 adapter ring is much thicker to move the sensor again away. So that is actually corrected. This focus should not make a difference. Doesn't matter with the T2 ring, because the T2 ring should always make sure that the distance of the sensor is correct. Okay, it's not only connecting the camera, but also it should adjust the distance. So what I'm gonna do now, it's 1.5 hours again. Okay, so just for the fun of it, last one under the, that's the cross section of an earthworm, and with that I think I'm going to call it quits for today. Again, where is it? Okay, yes, so this here is an earthworm. I got sneeze. Okay, excuse me. For whatever, see, I have to rotate the ring here a little bit. Yeah, here we go. Yeah. So that is, an cross section of an earthworm. Also a nice, and relates, it goes back a little bit to the previous questions, is can you see the cells? Well, yeah, you'd kind of have to zoom in quite a bit, and because they're so tightly connected, you might not see them as different anymore, because they don't have a cell wall. Yeah? So that is the thing here. Last comparison slide. Okay. Yeah, I have to go down with the image and refocus a little bit. And flip out the condenser. Here we go. Yeah, you do see that it's quite a bit more sharp here. Yeah. So what I'm gonna do now, people, what I'm gonna do now, people, is I think I'm going to call it a day again. Just wanted to do a little comparison today. If you have any questions about anything, please feel free to ask. Of course, if you have any suggestions for some videos, please also ask. There's been the request that I mix some horseradish juice with some of the nematodes to see what they do. Okay, why not? Why not try that out a little bit? Maybe I'm going to think about not only adding horseradish, but maybe also garlic and some other interesting spicy food. And let's see how the microorganisms and some of the worms that I have here in my jar, how they react to it. So maybe that's something I'm gonna do next week. I don't know yet. In any case, let me quickly see what other questions there are. How small should the hole be in the modification of the... Okay, how small? Honestly, I've tried everything already. You experiment around with it, but I've experimented with the size of a needle. If you try to make a nice round hole with the size of a needle, then you're able to see a significant difference already, but don't be surprised if the image is going to be quite a bit darker. And centering the hole is also important. Okay, so you need a little bit of patience for that. But what you can do is you can, of course, try to make holes of a different size and glue them over the different other holes, and then you have something almost like a condenser where you can, by rotating it, also change the aperture around. Lots of possibilities for experimentation, right? But there is I'm just gonna give it another one here. Try to avoid an adapter with between the camera and the lens. Use a camera from Bresser, and there is nothing between the sensor and the lens. That's possible as well, but you need to be able. So the suggestion has been is to use an adapter which directly. Without any optics, which directly projects the image from the objective directly into the camera sensor. This is sometimes possible, I found out, but it depends also a little bit on the size of the sensor camera and also on the distance of the tube and so on. So plenty of things to experiment around. Hope that you like the live stream. Hope to see you again. I wish you all the best. If you're interested in some of my previous videos, then please do check the channel, because some of the things that I talked about also made references to my previous videos. Okay, bye-bye everyone. Happy microbe hunting. See you around next time again. All the best.